Okay, I think we probably should get started. My name is Sharon Reichart, and I'm a member of the local Brain Network Support Group here in the Bay Area, and also on the board of a newly formed nonprofit, which is also called Brain Support Network, which is to assist people in the brain donation process. And I think Robin hit on it a little bit today, and we're going to be talking about it again later on this afternoon. My husband, Dennis, was diagnosed in 2002 with PSP. At that time, he was 58 years old. He passed away in August of 2007. And during that time, Dennis participated in UCSF's study and donated his brain to UCSF upon his death. And at autopsy, it was confirmed PSP. During those years, we were very lucky to be under the care of Dr. Boxer and his team, who guided us through our long journey. And I have nothing but admiration and praise for Dr. Boxer and the team for all they are doing in their search for answers for the cause and cure of PSP. Dr. Boxer will speak today for about 20 minutes and answer questions for about 20 minutes, and at which time he will need to leave. Please write questions down for Dr. Boxer on the cards that are on your table. Hold the card up and they'll be collected, and we will then have a chance to present the questions to Dr. Boxer. After Dr. Boxer, we will hear from Lisa Voltarelli, who is UCSF's research coordinator, and she will talk about PSP research and participation. And finally, if there's time remaining, we'll hear from Phil Myers on 8 lessons learned from PSP. So at this time, I'd like to introduce Dr. Boxer from UCSF's Memory and Aging Clinic. Dr. Boxer is the principal investigator of the worldwide Davunitide clinical trial in PSP. He is Associate Professor of Neurology at UCSF, Director of Neuroscience Clinical Research Unit, Director AD FTD Clinical Trials Program. Let's give him a warm welcome.
Thanks. Thanks, Sharon. Can everyone hear me? Is this working? Okay. So I wanted to thank Sharon and Robin Riddle and all of you for inviting me to speak to you today. It's really a great honor, and I just want to apologize that I have to leave a little bit early, but Lisa Voltarelli, who works with me in running our study, will be available to answer questions, and if there are any she can't answer at the end, that I'd be happy to give you a call afterwards and answer any that aren't fully answered today. So what I want to talk to you about is the research that I'm really focused on, which is really trying to find a cure for PSP. Everything that we do in my research group and with the research studies we're involved in are sort of focused on developing new treatments for PSP. And as you know, there really aren't very many good treatments that are available right now that can really affect the underlying course of the disease. And we've been spending a number of years trying to think of ways that we could really attack the underlying biology of the disease, both scientific experiments, but also working with the government and with the pharmaceutical industry to really increase the amount of work done in PSP. So I just want to give you an overview of how we think about this and the type of work we're doing. And first of all, I think everyone in this room probably has a fairly good sense of what the clinical disease PSP looks like for most people. And it was first described by Steele, Richardson, and Olszewski in 1964. And I think that their title of their paper, Progressive Supranuclear Palsy, a Heterogeneous Degeneration Involving the Brainstem, Basal Ganglia, Cerebellum with Vertical Supranuclear Gaze, which is the eye movement abnormality, Pseudobulbar palsy, knuckle dystonia, which is the neck rigidity, and dementia, which sometimes occurs with this disease, still holds today for many people who have the disease. And what we've learned, let's see, is this a pointer or no? So what we've learned over the years is that really PSP is very strongly associated with a protein called tau. And as we see it, PSP is really the archetypical disease that's caused by this protein tau. Tau is also found in many other neurological diseases, most notably Alzheimer's disease. But in PSP, there are many reasons that we know that it's very strongly linked to the disease. And if you look at autopsy, what you see is that in this picture here, this colored area is where the tau is deposited in the brain in most people who have PSP. And so the highest concentrations are in this brain stem, or the lower part of the brain that controls swallowing and neck rigidity and movement, but there are also some tau throughout the higher parts of the brain that involve thinking. And usually it's in certain types of cells. Here is a picture of one called a tufted astrocyte, which is filled with tau. And so our basic scientist colleagues have been thinking about how to treat different neurological diseases. And one of the interesting discoveries that happened about in 2007 by some colleagues at UCSF was that when they were trying to find a treatment for Alzheimer's disease, they knocked the tau protein out of these Alzheimer's mice, and they found that if they did this, so an average mouse with Alzheimer's disease will die pretty quickly in about 6 months, but if they take out the tau protein, they survive and they do much better. And they also found that if they take out this tau protein and expose the mice to toxins that are called seizures, they can also protect the mice from seizure-induced brain damage. So it appears that tau is really a central player in most neurological diseases, even though we don't see it always deposited or in the brainstem like we see it in PSP. And so this is really one of the key arguments that we're making to the federal government, to the NIH, to the FDA, to the pharmaceutical industry, that they should be focusing on tau, and particularly they should be focusing on PSP, because if you cure PSP, you're going to cure a huge number of brain diseases. One of the other really interesting advances in the past couple years, and just within the past year, it's become very apparent that tau normally in the brain is a protein that helps stabilize the sort of tree-like structure of a nerve cell. So if you've seen these beautiful pictures of neurons that look like trees with all the different branches, inside the branches are something called microtubules, and the microtubules remain stable because of the tau protein. And when the tau protein is dysfunctional, it starts to stick together, and the microtubules start to fall apart, and that's when you see the cells start to shrivel up. And it's really in a large part due to the dysfunction or loss of normal function and sticking together of the tau protein. What we've learned in the past year or two, that not only can the tau protein come off the microtubules and cause the cells to shrink and die, but it can also move from one cell to another. So at the bottom here, this just sort of summarizes that what we've learned importantly, that the tau protein can go from one sick cell to another. And that can probably cause the disease to spread throughout the brain. And this is important because we could potentially block the transmission of one cell to another. And some of the treatments that I'm going to tell you about probably do this. So over the past 10 or 20 years, people have been thinking quite a bit about how we could treat tau-related neurodegeneration. And here's a picture of a nerve cell like I just described. And these lines here are the microtubules with the tau protein. And all of the names that we're talking about here are different approaches as to how we might make this tau-related nerve dysfunction better. So one way might be to just stabilize the microtubules, and even if the tau is dysfunctional and falling off, if we were just to keep them in place, the cells wouldn't shrink and die. And so the drug davunitide, the study that Sharon mentioned earlier, is a drug that we think acts by doing this, that it helps maintain the stability of the microtubules, and we think that, or we hope that that will help to maintain people's quality of life. But there are a variety of other drugs that I don't have time to go into right here, but would be happy to talk about later. So as we try and convince the drug companies who have these drugs to use them in PSP, we make the following argument, that Even though most of the tau drugs were initially intended for use in Alzheimer's disease because that's going to make more money for drug companies, that we really need to convince the industry to invest in PSP. And the reasons that they might want to do this is that PSP is purely a tau disease. It's about as purely tau as any disease could be, genetically, neuropathologically, and otherwise. We know that patients have no other really effective treatments, so there's really a lot of incentive for patients to participate in clinical trials and to work on developing these therapies. And then also we know that more and more tau is associated with other common diseases, including chronic traumatic encephalopathy that affects NFL players and veterans coming back from the wars. And so again, if we could cure PSP, we could cure many other diseases. This is just an example of some of the other neurodegenerative diseases and the proteins that are present. And what we know is that particularly PSP, but some of the other disorders related to it are really purely tau, whereas these other diseases like Alzheimer's disease or dementia with Lewy bodies have multiple toxic proteins that are present. And that makes it very hard to demonstrate that a drug that's targeting tau is really going to work in this population. So I'm going to skip this slide because of time. And I want to tell you a little bit about the challenges now. So we're making a really big effort to get these drugs and to test them and develop them for PSP patients. But we have to acknowledge that there's some big challenges. And first of all, we need our scientists to work on the pipeline, and that is they need to develop more chemicals and compounds that could actually be tested and efficacious. We need better animal models. So there are mouse models that develop tau-related degeneration in the brain, but none of them look exactly like PSP. And so that's another thing that we really need to work on. We know that PSP is more rare than Alzheimer's disease, and so we need to really work on raising awareness and getting people to participate in research so that we can have enough patients to really develop these medications in clinical trials. And really one of probably the biggest problem with developing effective therapies is that many PSP patients aren't diagnosed until they're pretty advanced in the course of their disease. So they see one doctor after another who doesn't really understand what this is, and then by the time the diagnosis is made, it may be difficult to really have an impact on curing it. And so to make this a little bit clearer, what we know is that the typical PSP diagnosis that I described to you and that was described in the 60s is really focused, derived from having these classic eye movement abnormalities, falls, neck rigidity, trouble with swallowing, and sometimes behavior and thinking problems. And we know that when someone has these constellation of symptoms, what we call probable PSP, that Dr. Litvin and colleagues initially defined in 1996, that if we look at their brain at autopsy, that it's almost 95% or 100% likely that they are going to have pure tau-related brain disease. But as we learn more about this disease, we're starting to recognize that there are multiple forms of PSP, or multiple ways that PSP can begin before it really meets these classic definition of PSP. So some patients may begin as sort of the classic falls and eye movement abnormalities, but more and more we're recognizing that other patients may actually look like they have Parkinson's disease initially, and they may even respond to Parkinson's medications for a year or two, and then they start to look more like PSP. Others may have just a walking problem with very little else for a number of years, and that's called pure akinesia and gait freezing. Others may have the corticobasal syndrome that Dr. Litvin probably described earlier today. We're also learning that some PSP patients early on actually don't have any movement problem at all. They have either trouble with language or they have trouble with behavior, and that this can predate any of the movement problems for a number of years. And so this is another way that PSP can begin. And finally, what we're learning is that maybe even before any of these symptoms, some patients will have what we call a prodrome. So they may be a little depressed, they may have trouble functioning in their job, and this, what we call psychiatric or dysexecutive prodrome, may actually be the earliest symptoms, and it may predate sort of this diagnosis by 5 years, as much as 5 years. And so really our challenge now is to try and identify patients in these earlier stages when they are more likely to respond to one of these treatments. And I think, as many of you know, depending on what the syndrome is initially, they may be seen by different doctors. And so what we really need to do is educate all of these doctors about all of these different possible ways that PSP can begin. And so that they can recognize it earlier and get the patients to proper therapies. So one major effort that's underway right now is to actually rewrite and revise the PSP diagnostic criteria that Dr. Litvin led in, over 10 years ago, now almost 20 years ago, to try and acknowledge these other presentations and find an earlier definition of PSP that we can use to develop treatments. The other thing that we're doing is trying to develop biomarkers. And what are biomarkers? Biomarkers are biological measurements that we can use to better diagnose more accurately and more precisely follow the course of the disease. And so different types of biomarkers are really one of the major efforts of research that are underway in the field right now. So one type of biomarker might be a brain scan. Others are body fluid measurements, like looking at the spinal fluid. And we think that using these biomarkers, we could go back, and if we had a good biomarker, look at these very early patients and use, let's say, a brain scan or a spinal fluid measurement to confirm at the earliest stages that these people have PSP, and then use one of these new treatments to hopefully have a major impact. So the biggest biomarker study that's underway right now is one that I'm leading called the 4-repeat tauopathy neuroimaging initiative. And this is going on in San Francisco at UCSF and also in 3 other places around the country. Actually, one is in Toronto. So Carmela Tartaglia in Toronto is leading a site, Brad Dickerson at Harvard at Mass General Hospital, and Dr. Litvin actually is our newest site who will hopefully soon begin enrolling patients. And I know that some of the people actually in this room are participating in the study or have thought about participating. So just to tell you a little bit more about it, and this is also the study that Lisa Voltarelli is organizing for us. What we do is we follow people with either PSP or CBD, and we know that at autopsy that they have what we call pure 4-repeat tau. That's why this is called the 4-repeat tauopathy neuroimaging initiative. And we look at different types of brain MRI scans. We look at structure. We look at blood flow. We look at the white matter, how the neurons are extending throughout the brain. And we look at other types of measurements, including spinal fluid, blood, and urine. And as well as we measure eye movements. And we do this every 6 months for a year. And then we look at which of these measurements are really going to be most accurate in diagnosing, but also capturing disease progression so that we could use them in a clinical trial. And others have really worked on this, and so what we can do is we can look at specific areas of the brain. What we find is that when we use these types of very detailed MRI measurements, we're much more sensitive and specific in our diagnosis, and it's much easier to demonstrate that a drug might be effective than if we just used a rating scale that we would do at the bedside. So that's really a major focus of this project. Some of the early data that we're getting out of the project, here's an example. So we measure eye movements. We know that in the typical PSP that it's called a progressive supranuclear palsy of eye movements, which means that it gets harder and harder to move your eyes up and down and eventually left and right. And if we just measure periodically how well someone can look up or down, what we see is that this gets worse very rapidly. And obviously, I don't need to tell anyone in the room who's seen this disease that this is a big problem with the disease. But when we're trying to develop a new therapy, we need a measurement to follow that could tell us that the therapy is working. And this may be one of them, and we've actually incorporated this into the Davunitide trial. A new measurement that's coming out of our Fortney study, and I apologize there was no slide in the handout for this because I added it this morning, is looking at the brain network using what we call functional MRI. And what we can see is that there actually is a PSP-related brain network, even in normal, young, and older people, and that it's possible that the disease spreads along this network. And if we compare PSP patients with normal older people, that within this network, the connections between the different neurons are actually reduced. And this may tell us a lot about the disease, and particularly when we have therapies that can block the spread from one area to another, this may be the way to really measure that. Another type of technology we're working on in collaboration with others at UCLA and other institutions is what's called a PET scan. And this is a radioactive scan with a dye that actually detects this toxic tau protein. And what we can see here is that in PSP, that particularly in these areas in the brainstem and in the basal ganglia that control movement, that there's a huge amount of this tau protein that we can detect with this new technology. And that the amount of protein, this tau protein detected with the technology is strongly correlated with the disease severity. And so this might be another way that we could show that a drug is working, because if the amount of this tau protein reduced using the PET scan, then we would really have a good idea that a drug might be really attacking the underlying biology of the disease. So the point I want to make is that we are really hoping that we can convince you and others to participate in this research, because the more people we have who participate, the better we're able to develop these methods, and eventually the more drugs we'll be able to bring into treatment studies and really get this disease effectively treated sooner. And so although this study that Lisa will tell you more about does not have a treatment right now that's part of it, the more information we collect, the more likely we are able to get new treatments. So I want to just switch gears and tell you about what's available, what's been tested so far, and just to tell you that you really can do clinical trials in this disease. So the largest clinical trial that was done in PSP was with a drug called Riluzole. It was called the NIP study. It was done in Europe, and that was completed in 2009. And Riluzole is a drug that's used for Lou Gehrig's disease, and basically it was found that it does not work for PSP or MSA in this therapy, but in this trial. But it did show that we could do clinical trials in PSP. Another therapy that's being studied is coenzyme Q10, and this is work from my good friend and collaborator Günter Höglener in Munich, and they showed that coenzyme Q10 in a small study had some modest benefits. And there are a variety of different investigators now working on these different what we call mitochondrial therapies for PSP. And mitochondria are the parts of the cells that create energy and help the cell to really function. And so it may be that improving the function of mitochondria may be a way to cure PSP. The next type of drug that's been tested recently are drugs that block an enzyme that adds phosphate groups to proteins. And we know that the tau protein has a lot of phosphate molecules on it, and this causes the protein to change shape and fall off the microtubules, and it's a key part of the disease in PSP. So one of the drugs that was developed is called this GSK3 beta is a type of enzyme that adds phosphate to tau, and there was a recent clinical trial of a drug called tidaglosib that was done in PSP, and unfortunately it was found that it did not work in terms of treating the symptoms of the disease when PSP patients were treated for a year. But interestingly, when they looked in a secondary analysis of brain scans, so those same methods that we're trying to develop as part of the Fortney study, they were able to see a slight benefit of the drug. And so again, it means that even though this particular form of the drug didn't work, there may be other more potent or better versions of this type of drug, and particularly if we combine them with better brain scans and new technologies, we may be able to really get them to work for PSP. I think another really exciting possibility, I told you about devunatide stabilizing microtubules, are a whole other class of microtubule stabilizers. One is called apothelone D, and this has actually just entered a clinical trial for Alzheimer's disease, and we hope that it will soon be in a clinical trial for PSP, but it's really up to the drug company that owns the drug to allow this to move forward. But this seems also to be a very interesting drug that really has very potently can stabilize these microtubules, and it has really pretty amazing effects in some of the animal models of tau-related neurodegeneration. Probably the last and probably most exciting from my perspective of the new technologies that are being developed to treat tau-related neurodegeneration, and we hope soon PSP, are what we call the immunotherapies. So like a vaccine or what we call a monoclonal antibody. And this is work from Eli Lilly, where they have mice that develop a severe tau-related neurodegeneration, and if they inject antibodies, so what happens is the mice lose weight and die very rapidly, but if they give them an antibody that binds to this tau protein, perhaps blocking the spread from cell to cell, the mice do a lot better, and if you look in their brains at autopsy, they have a lot less tau that develops. And there are many versions of these different tau immunotherapies that are being developed, and the first of them will be entering human clinical trials, hopefully in 6 months or less. So our one major effort that we're involved with is trying to convince some of the drug companies that they should not be wasting these antibodies in Alzheimer's disease. They should be using them to treat PSP first. So that's really something that I'm very interested in right now. We heard a bit about devunatide. That clinical trial is going on in about 320 patients at 47 centers around the world. We should have the results in late December or early January, and we hope that it works. If it does, then that could be the first drug to be approved to treat PSP. I won't go through the details of the study, but we're very hopeful, and even if the study unfortunately does not show that the drug works, one of the exciting things about it is that because we designed the study and arranged it at UCSF, we will get all of the data from the study and we'll be able to use it to refine our methods and develop better trials in the future. So I think with that, because time is limited, I'm going to stop, and this is just sort of a summary of what I talked about, and just open it up for questions.
Are there any cards to be handed in for questions? Yeah.
Yes.
Okay. My question has to do with dopamine and PSP. I understand that one of the ways to distinguish PSP from Parkinsonism is that it doesn't respond to the dopamine stimulators that seem to help in Parkinsonism. However, I believe Dr. Litvin said that all of these atypical Parkinsonisms have something to do with dopamine. And what I don't understand is, for instance, in our case, my wife has restless leg syndrome and is taking a drug for that which stimulates dopamine and is helping her restless legs. So why wouldn't the regular Parkinsonism type drugs also help her?
So that's a great question that we could talk about for a very long time. But so the question is, why don't drugs that elevate dopamine, some of the drugs that people use for Parkinson's disease, why don't they work for PSP? And so the answer is a little complicated, but Parkinson's disease and dementia with Lewy bodies and probably to some extent multisystem atrophy are caused by accumulation of this protein called synuclein, which is different than the tau that's in PSP. And one of the areas that it really seems to hit is this area in the brainstem called the substantia nigra that produces actually dopamine. And the dopamine goes along neurons and spritzes into the basal ganglia that control sort of automatic movement. And so when you lose that dopamine in Parkinson's disease, you get the tremor and the stiffness and the falls. And so replacing it in Parkinson's disease and to some extent DLB and sometimes multisystem atrophy makes people better. PSP does not really affect the substantia nigra nearly to the same extent, and there isn't this dopamine deficit. But that being said, it does affect the basal ganglia and other areas of the brainstem, and it may be that by giving back a little bit of extra dopamine, some PSP patients will get a small benefit for a short period of time from the dopamine. But ultimately, it's a very different disease chemically than the other Parkinsonian diseases, because dopamine is not the central problem. Does coenzyme Q10 work with the drugs? It can be taken, if that's the question, with the drugs. It works differently. So again, it helps to, we think, has a very modest effect on these mitochondria, which are in all of the cells in the brain, and helps them to create energy a little bit better. And there's some thinking that's with some of the Parkinson's drugs that they may stress out the mitochondria a little bit, so the coenzyme Q10 may help. But in general, I think there have been many studies now with coenzyme Q10 and other diseases, other brain diseases like Huntington's and Parkinson's, and it hasn't had a huge effect. There is an ongoing clinical trial in PSP, but so far it doesn't seem to have the type of effect that we'd really hope for in terms of curing the disease. So it's, if this approach is going to work, it's probably not just coenzyme Q10, but it's going to have to be a combination of coenzyme Q10 with other drugs that affect these energy producers within the cells. Can a patient be too advanced for research? So I think it depends on what type of research the patient and the family want to do. So in our research project that Lisa can tell you more about, one has to be able to tolerate the MRI scans and some of the testing that we do. Otherwise, we just can't get the measurements. CurePSP, the National Society actually has a brain bank, and I think Sharon, you might talk, you mentioned a bit about how you're working on an autopsy program. So we also have an autopsy program at UCSF, but in our autopsy program, we really value taking the clinical data and matching it up with the autopsy data. So, so for our programs, people can't be too advanced. They can be fairly advanced, but they have to be able to do the brain scans at the very least. But the CurePSP brain bank, I believe, will take any person's brain who had PSP, so even if they were very advanced. But if there's any question, we'd love to talk to you about it, and we'd love to work with you in any way that we can to allow you to participate in the research. Does anesthesia cause or exacerbate or bring on these diseases? And this is a major question, I think, that many people are interested in. And there are a variety of different hypotheses that explain why many people see this to be the case, that often the disease didn't really, maybe there were some little hints before they had surgery or anesthesia, but it didn't really get going until afterwards. And so one possibility is that the oldest explanation for this is that your brain actually is compensating for the disease for many years before you actually have symptoms. And so, the disease probably starts, you don't notice anything for years and years until enough of this tau protein builds up to cause the disease, because the rest of your brain is working overtime to make up for it. But when you have another injury or you have anesthesia, that may block some of this overtime working of the rest of the brain, and that may unmask the symptoms. That's one explanation. Another newer explanation that people have learned in the past 5 years or so, that inflammation, so actually the immune system in the brain is somehow activated with PSP and other related diseases, causing the release of all kinds of inflammatory chemicals that seem to make the disease worse. And it's a chicken and egg. We don't know whether it's the inflammation that starts the disease or the disease causes inflammation that then makes things worse. But what we do know is that any kind of surgery or injury, let's say you even have a bad infection, you may have orthopedic procedure on your leg, that creates inflammation that spreads throughout the body. These inflammatory chemicals gets into the brain and can make some of these neurological disorders even worse. So that's one possibility. A third possibility is rarely there are people who undergo surgery, let's say if for a cardiac procedure where they're on a bypass pump, and you can have either a small stroke from a clot or from an air bubble that can go into the brainstem and produce a syndrome that looks like PSP, but this usually doesn't get worse. It just stays stable. So I think that we don't know exactly the answer, but the idea that inflammation may be actually the real culprit here is exciting because it suggests that we could potentially prevent that from happening. So if you knew somebody was in the earliest stages of PSP, then perhaps you could give them some sort of anti-inflammatory medication or medication to block this effect that might worsen the disease, and that would be really an effective way to prevent the disease from really manifesting itself. And so there's quite a bit of research going on to look at that possibility.
Dr. Litvin hit on this a little bit, but we've got two people who've asked this question here. What are the latest research findings on heredity or genetic markers for PSP?
So CurePSP, the foundation just sponsored a really exciting, very large genetic study of PSP. And I think what I alluded to in the beginning is that we really think PSP is extremely tightly linked to this protein called tau. And it turns out that when they looked at over 1,000 brains that were confirmed by autopsy as having true PSP, and they asked which genes anywhere within the whole human genome were associated with getting PSP, and they found multiple hits within the tau gene, that actually tau is extremely strongly linked to getting PSP. So it may be the levels of tau or some other things about the genetic regulation of tau that's extremely important. It turns out that there are rarely within families people who have actual mutations in the tau gene, and that depending on where the mutation is in the gene, it can cause these PSP-related tau abnormalities, and they actually get a familial form of PSP. There were also a number of other genes that were found in this very large research study. Some of them actually had to do with inflammation. So we were just talking about how inflammation may be involved with PSP. So that's one other class of genes, and people are looking at that. Another class of genes that seem to be related to PSP that came out of this study are genes that are involved in other aspects of the neuron structure. So there is an insulation around the neurons so just like you have insulation around a wire, that if it's a metal wire with plastic, there's a similar type of insulation around neurons, and it turns out some of the genes for that insulation also appear to be related to PSP. But still strongly, I mean by far, more than anything else, this tau protein gene in many different ways appears to be the by far strongest link to PSP, and that's why I'm so focused on tau, and we're really trying to tell everyone else to focus on tau, because clearly there's no disease that's more strongly linked to tau than PSP. So how long would davunitide or another drug take to get into general use, let's say, if it's effective? I think it's hard to give you an exact answer. With davunitide, we worked closely with the Food and Drug Administration to have a special agreement with them. One of the advantages that PSP has, because it's considered to be an orphan disease, that is, there are fewer than 200,000 cases currently in the United States, is that the Food and Drug Administration has special programs to allow accelerated approvals for these types of drugs. So if we are, God willing, fingers crossed, successful with this davunitide trial, we could get what's called an accelerated approval, which means that, assuming the data really looks strongly positive, it might be, a matter of 3 to 6 months before we get FDA approval, and then it might be, really it's a question for the company that manufactures the drug, how quickly could they make it at that point. But I think if the study looks positive, then everybody will bend over backwards to get this drug made as quickly as possible.
Next question is about side effects. Are there side effects to davunitide?
So, I can talk about our experience. We did a small study with davunitide. I can't talk about the study that's underway, but that we did a small study that was funded in part by CurePSP in a small number of PSP and CBD patients. And what we found is that, and one of the reasons why we were enthusiastic about davunitide is it seems to have many fewer side effects than some of the other drugs that have been used in the past. But the biggest problem, I would say, and anyone who's been in the davunitide trial could probably agree with this, is that currently the only way to give the drug is as a nasal spray. And so for some people who so you have to take the spray 2 puffs, I think, in each nostril twice daily. And that can cause some irritation to the nose or some congestion that can be a problem for some patients, because if you have swallowing problems already, having your nose clogged up is not very pleasant. So that's the biggest side effect. I think there have been some other minor side effects that we saw, like headache, sleepiness, but no, we have not seen any serious side effects with davunitide.
If you have more time, we have more questions.
One more question?
Okay, one more question. Does L-DOPA help PSP?
So traditionally we say that L-DOPA does not help PSP patients, and that's sort of been one definition of an atypical Parkinson's disorder. That is that unlike Parkinson's disease, where L-DOPA really makes a difference, the atypical Parkinson disorders like PSP don't really respond to L-DOPA. But what we know is that there are a percentage of PSP patients who do have a benefit from L-DOPA. Usually, though, it doesn't last. It may be for the first year or two, and as I mentioned, there is now recognized a form of PSP called PSP Parkinson's disease, or PSP Parkinsonism, and those patients really look like they have Parkinson's disease, even to the point that they respond to L-DOPA initially, but that usually goes away after a year or two, and then they start to look more like PSP patients. So what we usually say is that anyone who has PSP should have a trial of L-DOPA or one of the drugs like it, because there is a small chance that it will benefit them. We have to have modest expectations, though, because we know that in most cases it doesn't last very long. So thank you. I'm going to invite Lisa to come up and sort of finish answering questions. And thank you very much for your attention. I apologize for the so longer.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.